When we are ready, Ms. Richardson.
I'm ready.
All right, thank you. All right. The rest of you ready? Ms. Richardson's ready. The rest of you ready?
Y'all ready?
All right.
Girls?
I'm ready.
Cell-mediated immunity. We're going to have two types of our third line of defense. Cell-mediated immunity and humoral immunity, or antibody-mediated. All right? So right now we're talking about cell-mediated. This is T cells. T cells. An overview, how it works. You're actually seeing an overview of how both types work. But we're right here in the beige right now. So we have a new antigen come on the scene. Our nonspecific defenses come into play. Remember, they work hand in hand. All right? This antigen here happens to be presented to an antigen-presenting cell. This is a dendritic cell. It is going to take that antigen and present it to a T cell. Okay, so it's going to activate the T cell. You see this as a naive T cell. Naive means a T cell that's been activated against its antigen, but has not acted against any of those antigens yet. All right? So this is what's happening. This cell is ultimately going to become a cytotoxic T cell. We're also going to form some memory cells that are held back. You see here, I'm skipping ahead a little bit, but we also have another kind of T cell. This is a helper T cell. They form some memories too, and so if we ever meet this antigen again, the memories are if we ever meet this antigen again, we're ready to act against it. I'm going the wrong way, excuse me. I'm messing up my markers. Let's go this way. Antigen is presented to this T cell. It's activated. It becomes a naive T cell. Some of the cells become memory cells. They're held back. This goes on to be a cytotoxic T cell. And it's going to go up here and act against this antigen. We are also sensitizing some helper T cells to this same antigen. Oftentimes, a helper T cell is needed to activate a cytotoxic T cell. So we're going to find out that helper T cells play a very important role. We also make some memory helper T cells. Nonspecific inflammation is stimulated. Now you can see here that this arrow from these helper T cells is also pointing over here onto the antibody-mediated side. We'll talk about that later. Helper T cells help both cell-mediated and humoral immunity. Very important. Helper T cells. Now, these are the cells of cell-mediated immunity. The cytotoxic T cells, which are most of them, about 80% of them, these are the cells that attack cells that contain the foreign antigen that they are specific for. Helper T cells are those that help activate those and stimulate those, both T's and B's. Regulator T cells are those that put the brakes on, so to speak. They are formed in lesser numbers and formed later than the cytotoxic T cells, but they will put the brakes on the immune response. Antigen-presenting cells are not T cells, but they are part of the cells that are involved with cell-mediated immunity. Antigen-presenting cells are those that present antigens to the T's and the B's. When we talk about cell-mediated immunity, we're talking about these things right here. We've got to recognize the antigen, and we use antigen-presenting cells largely for that, but not exclusively. The T cells have certain identifying markers. These are called CD markers, or cluster of differentiation markers. Cytotoxic T cells have different markers from helper T cells. And then we have a process called co-stimulation. This is basically a safety check mechanism, so that once the T cell is activated and ready to go to work, there is a built-in backup safety check mechanism to make sure that this T cell is going to attack a foreign antigen contained in certain normal body cells. So we don't want this T cell attacking normal cells. We want the T cell to only attack where the antigen is. And that process is called co-stimulation. Now, how do T cells recognize antigens? There are two classes of specialized proteins called MHC proteins, major histocompatibility complex. There's type 1, class 1, and class 2. Class 1 is a type of protein that presents fragments of the antigen on the membrane of infected cells. So cells that contain the foreign antigen, that antigen is broken down internally, and a fragment, not the whole antigen, but just a fragment of the antigen is placed on the cell membrane, so that if and when the T cell community, these single or multiple, come by that are specific for this antigen, they will recognize it and will be stimulated to go ahead and attack any cell in the body that contains this fragment. So it's amazing that the T cells can recognize even a fragment of the total antigen. But this is what happens in fixed nucleated tissue cells. And so class 1 MHC proteins. All right? Class 2 MHC proteins are those involving the antigen-presenting cells. These antigen-presenting cells move around. All right? And so they can pick up the same antigen that you just saw in that fixed tissue cell for class 1. So they phagocytize the antigen, break it down internally, and present a fragment of it on their cell membrane. And they can take that fragment of the antigen and place it to places where there are lymphocytes. Where do we find a lot of lymphocytes?
The lymph nodes.
In lymph nodes.
Mm-hmm.
So they can take the lymph nodes so that the lymphocytes that are specific against this antigen can recognize it, be activated, clone themselves, and go after it. So antigen-presenting cells can recognize the same antigen that can be found inside fixed tissue cells. This is a little comparison of class 1 and class 2 type MHC proteins. All right, so all nucleated cells, class 1. Antigen-presenting cells, class 2. Recognized by CD8 cells, these will be the cytotoxic T cells, and also by the over here by CD4. Foreign antigens that appear inside cells, these are called endogenous antigens. Those that appear in body fluids outside of tissue cells are called exogenous. So if we have a foreign antigen on an MHC protein here in class 1, if it's displayed by an antigen-presenting cell, it says, I belong to self, but have captured a foreign invader. This is what it looks like. Kill any cell that displays it. If it's displayed by any other body cell, it says, I belong to self, but have been invaded by something cancerous or foreign. Kill me. Now, over here, MH2, I belong to self, but have captured a foreign invader. This is what it looks like. Help me mount a defense against it. Now, why is it important for the antigenic fragment to be presented on the cell membrane for the T cells? Because T cells can't see inside a cell. It has to be on the surface of the cell so they can see it. These antigen-presenting cells, what kind of cells are we talking about? Well, they're these right here. Dendritic cells, macrophages, and B cells. Later I'll show you why a B cell is classified as an antigen-presenting cell. Here's an electron photomicrograph of a dendritic cell. Here's an electron photomicrograph of a macrophage enveloping bacteria. See the cytoplasmic extensions here. Here is a B cell that has identified its foreign antigen. Here is a helper T cell that's come along to co-stimulate it. So this is why B cells are in this class of antigen-presenting cells, because B cells present their antigens to helper T cells. These markers on cells. There are a number of different kinds of markers, CD markers, but two prominent ones are CD8 and CD4. All of the cytotoxic cells have a CD8 marker. All the helper T cells have a CD4 marker. This is a little marker on the membrane of the cell. So here we have our immature lymphocytes in red bone marrow. They're going to, if they're T cells, they're going to pass through the thymus, aren't they? And they're going to be programmed. They're going to become immunocompetent. So both the CD8 marked cells and the CD4 marked cells pass through here. And this just shows you the progression. They're cloning themselves, making some memory cells. These are cytotoxic cells. The helpers are doing the same thing, cloning themselves, making some memories, and these are the active helper T cells here. Now in this case, in this little diagram here, the foreign antigen is being presented by a dendritic cell in both sides here. As I said, co-stimulation is just a safety check mechanism to ensure that the T cells do not attack normal, healthy self cells. Now, in nucleated cells, an example I showed you earlier, the T cell will recognize the antigenic fragment on the cell membrane, but it must also bind to another site on the membrane prior to activation. That was not shown in that diagram, but it happens. In body fluids, not in fixed nucleated cells, the T cell binds to the antigen-presenting cell prior to activation, but it usually also requires a helper T cell. So helper T cells are important in both B cell activity and T cell activity. I'll stress this again later. This is what I was telling you. I showed you this a while ago. Here's the antigenic fragment. This is class 1 MHC, endogenous antigen. This T cell that comes along as specific for this antigen, it's not activated fully just by contacting this antigen. It has to bind to another place on this membrane to be sure that it's going to attack, not going to attack normal cells. And here's an example of helper T cell assistance with a cytotoxic T cell. So in this case, the antigen was presented to the T cell right here, but we need the help of a helper cell here. Because this helper cell is going to come along and is going to release some specialized immune proteins that are going to co-stimulate this whole process and allow this cell to become fully activated. If this doesn't happen up here, this cell is going to be tolerant to that antigen.
So, doctor, there's always a helper cell in with the T cell, in and around the T cell to come to them, help them.
Not always. In body fluids, where we have antigen-presenting cells, there's always a helper T cell. In fixed nucleated tissues, where we, in organs and in body tissues, you don't always need a helper T cell.
Okay.
All right, so it's just a safety backup to make sure that these T cells don't attack normal cells. All right, so let's go over the cells. Cytotoxic T cells. They can respond to both types of MHC proteins. They attack any cell that contains the foreign antigen that they are specific for. As I told you before, most of our T cells are the cytotoxic variety. They also stimulate inflammation and natural killer cells. How do they kill? They kill by creating holes in the membranes of those cells that contain the foreign antigen, and they release enzymes in that actually break it down and kill it. So here would be our T cell. This would be a cell that contained the foreign antigen. Here it's perforating, making holes in the cell membrane and releasing these enzymes here. Here is an electron photomicrograph of a cytotoxic T cell acting against the cancer cell in just this fashion right here. All right, then we have our memory cells. They're held in reserve. They remember the antigen. If after this first episode that same antigen is encountered again, we already have a community of T cells that are specific against that antigen, so the reaction is going to be faster and stronger than the first time. So here are our memory cells. The regulator T cells are the ones I told you that come along later in lower numbers and essentially put the brakes on the immune response. Here's where they're classified right here, regulatory T cells. Now the helper T cells. Notice that we say that these are involved in both cell-mediated and humoral immunity. Their major function is stimulation of proliferation of other T cells and B cells that have already become bound to antigen. Without helper T cells, there can be no effective immune response, because they're involved so intimately in the whole process. So here's the helpers over here. So, as far as T cells are concerned, what do? They provide co-stimulation, they help activate the cytotoxic cells, and they promote proliferation of more of the same. What about B cells? Basically, they're doing the same thing. Co-stimulation, as you saw, promoting proliferation of B cells, stimulating production of antibodies. And besides that, they activate macrophages, natural killer cells, and stimulate inflammatory response. So if you don't have helper T cells, you can't have an effective immune response. This is what happens in HIV/AIDS. The problem is with the helper T cells. Their numbers progressively decrease, and as they do, the situation gets worse and worse. This again is just kind of a repeat of what we're showing. I've shown before. The beige down here represents T cells. So here's an antigen-presenting cell, presenting the antigen to the T cell. Here's a helper T cell coming alongside to co-stimulate and expedite, facilitate the activation of this cytotoxic T cell against the antigen it's specific for. Up here, you see the B cell presenting its antigen to a helper T. And helper T is providing co-stimulation for this B cell. This B cell, once this co-stimulation process is complete, it will go on, clone itself, make plasma cells, which make the antibodies. Now, a little bit about homograft rejection and immunosuppression. So what we're talking about here is tissue and organ transplants. Now, so the basic problem here is dealing with self versus non-self antigens. So let's say somebody has a kidney transplant from other than an identical twin. Right now, lots of people have had this, right? And most of them are successful, aren't they? All right. How is that so? Because an organ from a non-identical person is being transplanted into this recipient here. How come the recipient doesn't reject it via its own immune system? Because these are foreign proteins, aren't they? Foreign to the host, to the recipient.
Yeah.
So how come the recipient doesn't reject this kidney transplant?
Same bloodline.
They're on immunosuppression for a while.
The immune system is suppressed, right? Now, most of the time that works, but sometimes it doesn't. Sometimes the recipient will reject the organ anyway. All right? So the answer, sort of, is immunosuppression. Now, is this just immunosuppression at the time of the transplant and it's done for life? No, it's an ongoing thing, isn't it? All right. So it's a two-edged sword, isn't it? Because if you suppress the immune system to allow the donated organ to survive,
So ignore all
are
foreign
you
antigens.
suppressing just that?
No, all foreign antigens.
You're actually suppressing the entire immune system, aren't you? So, yeah.
I watched on Nova Science where they're doing, or they're, it's experimental, but they're taking, donor organs and they're stripping them down and they're putting the person's own cells on there to make them grow that way that there's no rejection.
Oh, interesting.
Something that we're trying to do.
But how do you keep that organ, quote unquote, alive while you do that?
It only takes like maybe 3 days or something like that to.
Maybe something sounds like they do with blood now.
Like what?
How they manage to do blood now, so they're trying to get everything to O positive.
Well, let's take an example of a person who's had a kidney transplant. And let's say it's been successful, has had no problem, has good renal function. But he now is showing up with skin cancers. Never had a skin cancer before we had the transplant. So we removed a couple of skin cancers. You see him back 3 months, 4 months. He's got another skin cancer or two. Remove those. Comes back 3 months later, got more. Why?
Immune system is
Can't
down.
fight off.
It's immunosuppressed.
Yep.
It's immunosuppressed. All right, some terms. Autograft. This is a tissue transfer from one part of a person's body to another part. His own tissue, or her own tissue. Let's say you got a burn of the forearm. It's full thickness. You need a skin graft. So you can go to another part of the body, like the thigh, and take a thin layer of skin, not the full thickness. Take what's called a split thickness skin graft. You put it here after the wound's prepared, and that graft will take here and cover the wound. Where you take the skin from, since you didn't take the full thickness, it grows back from the accessory organs of the skin. All right? An isograph is a transfer from one person to another that have the same genetic makeup
Like
as
twins.
in identical twins. An allograft, also known as a homograph, is a transfer from one member of a species to another member of the same species. All right, so, for example, a burn victim, major burn victim, got multiple wounds that need to be covered temporarily. You go to a skin bank and get donor skin to put on those wounds as temporary cover. Now, will that skin take? Well, it might take initially, but is it going to be there permanently? No, because it's not of the same genetic makeup, so the recipient's going to reject it. But in the meantime, you bought some time. You prevented protein loss. You kept down infection until such time as you could cover those wounds with the patient's own skin. Another way of doing the same thing is with a xenograft, also known as a heterograft. You got a major burn wound. You debride this. You've got open wound. The main problem, if a person survives the hemodynamic insult of a major burn, long term after that is protein loss and infection, sepsis. So you got to clean the wounds up and cover them. And say you don't have enough skin to cover everything. You can buy some time by putting pig skin on these wounds. Porcine graft is from a pig, so it's from another species, isn't it? All right. This does a very nice job of cleaning up the wound, preventing protein loss, holding down infection, and it's amazing how clean you can get a wound looking just by this. Now, how long do these porcine grafts last? 2 weeks, maybe max 3, but then they're going to come off. And you can clean them up, put some more on, but eventually you're going to cover it with the patient's own skin.
We watched a video in my anatomy 1 class where they had, it was like with that, but so that people don't get an infection, I know they gotta cover the skin, but you can't like put rapid or anything. They had this thing where they, I guess it was kind of like a spray paint thing, but they had created this system where they could, I guess like make skin, and they would spray it on to the patient. It's like some new technology.
Well, one thing that can be done is you can take skin, normal skin from a burn victim and put it in culture. And,
Yeah, I think that's what they were doing.
You
They
can
were
grow
like spray
epidermal
painting
cells.
on.
You can grow epidermal cells and put those on the wound.
Yeah, we watched the video. That was pretty neat.
Now, there are other similar techniques to mix skin and put a kind of a synthetic skin on there. But ultimately, it's going to have to be covered with the patient's own skin.
It was really cool.
All right, antibody-mediated immunity. When my watch hands get a certain way, I can't see if I see it now. All right. Now we're over here on the green side. Now notice this antigen, this new antigen here, was recognized by T cells. The same antigen can be recognized by B cells. Okay. Now notice that we're going to need our helper T cells here. And those helper T cells are going to co-stimulate, going to activate, and this B cell, once activated, is going to clone itself. It's going to make some memory cells, hold them in reserve. These plasma cells come from B cells. The plasma cells are the ones that actually make the antibodies. So we're talking about antibodies now that are acting against this antigen wherever it appears in body fluids. Our T cells act by killing any cell that contains the foreign antigen. Antibodies don't kill, they immobilize, but they act against the same antigen.
So in theory, the antigen actually turned into an antibody by the end of the whole process. What started
No,
as
no,
an
no,
antigen,
no.
no? Okay.
Antibody is the body's defense against the antigen.
So it kind of absorbed the antigen?
Once it immobilizes it, what does it do?
Right,
I'm gonna show you.
okay. I think that's what I'm missing.
I showed you this before.
Uh-huh.
This is the B cell having recognized the antigen is specific for. Here's the helper C cell that's coming alongside and co-stimulating and activating this B cell. This B cell is going to go on to clone itself, make plasma cells, which make the antibodies. But if we don't have good helper T cells, we're not going to be able to activate these B cells. And so this B cell would be tolerant against its antigen, wouldn't it? If it's not fully activated and co-stimulated. So what are these antibodies looking for? They're looking for foreign antigens in body fluids. They don't usually get into solid tissue. Sometimes they can if there's a specific lesion. But mainly they're acting in extracellular fluids. Now, I told you that antibodies don't kill. You might have thought that antibodies do kill, but they don't. They immobilize their prey and set them up for destruction by other means, as I'll be showing you. So this is just a review of what we've said about how B cells work. All right, so here we've got our activated B cells cloning themselves. We've got some held back as memory cells, and these are going to produce the antibodies down here. Again, just showing you the assistance of the helper T cell and the B cell being activated. Now, what about the structure of an antibody? They have kind of a Y shape. So they have a C or stem region right here. And then they have some segments that branch off like this. So you can see here that there are some heavy and light chains out here. But the important thing is that the specificity of an antibody resides out here in the terminal ends of these Y branches right here. So all antibodies are proteins. And these proteins have a certain shape. Shape determines function for proteins, doesn't it? So the shape of these antibodies out here on these terminal ends right here, these antigenic terminal sites here, have a very specific makeup, and they will only respond to the antigen that fits right against the ends of that antibody. Any other antigen won't act against it. So antibodies are specific against one particular antigen because they recognize only the specific molecular structure of the antigen that they are specific for. So as we said, all antibodies are proteins. We looked at the constant and variable segments. This is just showing you, for example, an antigen with different antigenic determinant sites. So antibodies don't envelop their antigens, they don't phagocytize them. They bind only to specific parts of them, called antigenic determinant sites. So these are the binding sites of an antibody. Now, this antibody here is specific only for this antigen. Down here we need another antibody because it's a different site. All right, but that's what we have. So, for example, when you have a transfusion reaction, you have clumping of red cells, don't you? And the configuration of the antibody is conducive to that because let's say that this was a red blood cell.
Okay.
And let's say that this was its surface antigen. So if this was type A blood, this would be surface antigen A, wouldn't it? All right. Now, this part of the antibody is, this part of the antibody could have touched there instead of this part, right? So if another red cell comes along here, it's going to bond to that, right? So, actually, I guess I should have this wrong. If this is type A blood as surface antigen A, so if it were transfused with type B blood has anti-A antibodies, right? All right, so it's attaching here. If we had another similar cell right over here, it would attach to that one, right? Okay, and so what you get is a cross-linking here. This is why red blood cells clump, because you can hook to one here, like this, and they'll clump. Antibodies are in classes. There are 5 different classes. IgM is the type that's first on the scene in response to any antigen, but it's not the most prevalent one. IgA is primarily in glandular secretions. These are antibodies like on mucous membranes that recognize antigens that come in the air or that we ingest. Okay. This is the way most of our antigens enter the body, right? We breathe them in or we ingest them. Right? And so it's our IgA antibodies that act against those. They're on the surface. They catch these things before they invade the tissues. So as I told you before, if you have an immunization that's injected into you with an injection, you're bypassing this class of antibodies here. IgDs are antigen receptors on B cells. The IgG stands for immunoglobulin, by the way. These are the most abundant and the main antibodies in our antigen-antibody reactions. IgE is the one that is involved with allergies. So this is active in allergic reactions. These are just some of the configurations of them. Now, we said that antibodies don't kill. They immobilize the cells that contain the antigen they're specific for, and those cells will be eliminated in some other way. So here are the ways. They can neutralize by blocking specific sites on viruses and bacteria. They can cause agglutination, as in clumping of red cells. They can cause precipitation, which is a similar reaction with chemicals in solution. Or they can be involved with complement fixation. Remember complement proteins? Complement proteins puncture holes in cells and kill them that way. So this is a pictorial review of what we just said. So here is how those antibodies work: by neutralization, agglutination, precipitation, or complement. Now, in any of these 3 ways right here, what happens? Phagocytosis and inflammation. Over here, however, if we have an antigen-antibody complex, that C region there makes way for complement protein to come in here and cause lysis of the cell. But antibodies themselves are not killing the cells. They're setting them up for elimination in some other way. Now, when we have an immune response with a new antigen that we have not encountered yet, the action is not immediate the way it is with our general defenses. Our general defenses will act immediately. But when we meet a new antigen as far as our immune system is concerned, there's a lag time while the antibodies are being built up. So we're talking about antibodies here. antigen-antibody reactions.
When you say while the antibodies are being built up, does it also include the cloning process?
All
Okay.
that's going
And the
on.
memorization, that's
It
all
takes-
happening?
It takes a few days.
Okay.
It takes a few days. Now, so look here. It's going to be about 1 to 2 weeks before we get peak antibody concentration. Because we've got to meet the antigen, we've got to be activated, we've got to be co-stimulated, we've got to be cloned, we've got to make the plasma cells, we've got to build up the antibody titer.
Okay.
And so it's going to be somewhere between 1 and 2 weeks before you get the maximum antibody response with the first encounter.
Okay.
That's what we're talking about right here. Now, once that happens, remember some memory cells were being formed too?
Mm-hmm.
So let's say we got through the first phase, we controlled the antigen, eliminated the threat. Those memory cells were held in reserve, but they recognize the same antigen. So let's say down the line, you meet the same antigen again. Bingo. You've already got a community of lymphocytes ready to go. The response is going to be almost immediate, and it's going to be much stronger.
Is that why there's always a greater concern for a new strain of a flu or a new strain of this or that? Okay.
That's right. Because this new virus is going to have a different molecular structure.
Okay.
So you're going to have a different community of antibodies.
Okay.
for that.
I have one more question going back to drawings when you were showing us how they were the antibodies were connecting to the antigens, and you got one branch of the Y was connected to the antigen, the other one's kind of hanging off, and you said another antigen could come up and it could connect to that. Does that antigen, when you showed us that the antigen had the 3 different things, do all 3 of those little antigens have to have an antibody connect to them for it to immobilize it, or could you have one hanging out there without any antibody?
Yeah, you just.
I know I'm kind of simplifying it with the graphic, but
But we made it very simple by showing you just one cell.
Okay.
But there are going to be more than one cell with that antigen in it, more than likely.
Right. But do they all have to have every little side grabbed onto? You can have little. Okay.
So here it is graphically.
Okay.
Here's our first exposure to a new antigen. It's somewhere between 1 and 2 weeks before we get a maximum titer.
Okay.
And let's say we respond to that, we control it. Down the line, we meet that same antigen again. This is our response now. But now here is a different antigen. So we have to have a different antibody, see?
Mm-hmm.
So, same thing, we got to go through the same thing. Now, most of the time, the immune response works normally. Sometimes things can go wrong. And so we have some autoimmune disorders. What is an autoimmune disorder?
Where your immune
It's
system doesn't-
basically failure to recognize self.
Yes.
So our immune cells end up attacking some part of our bodies. Now, why does this happen? Well, there are different, theories on this, but one is that some self-reactive T and B cells escaped elimination in the thymus and the bone marrow, respectively. Some slipped by. Possible. Maybe we encountered new self-antigens that were previously exposed to these B's and T's. Possible. Could be a case of mistaken identity. Sometimes there are body parts that have a molecular structure that's very similar to a foreign antigen. And so our immune cells will attack that tissue.
Okay.
For example, this is what is thought to happen in multiple sclerosis, because the makeup of the myelin sheaths is very similar to maybe a foreign antigen that a T or a B cell is supposed to be specific against, and they'll attack myelin because they've mistaken, self for non-self. Immunodeficiency diseases. Example, HIV/AIDS. What is the problem in HIV/AIDS that's causing this immunodeficiency?
Those helper
What
cells,
is a person with HIV deficient in? Helper
the helper.
T cells. Helper
And it's
T
because neither your T's or your B's can work without the helpers.
That's right. So that handout I gave you on HIV/AIDS, you read through that. The very last sheet of that is a little chart that shows you the natural history of HIV, which terminates in AIDS eventually. You've got some review questions that come off of that handout on HIV/AIDS. And you've also got the answers to the questions. So on your lecture test, there will be some questions on HIV/AIDS. But you got all the information that you need about HIV/AIDS in those handouts. All right. Here's another malfunction. Hypersensitivities. We're basically talking about allergies now. And there are different classes. We've got several classes. I'm just going to touch the highlights of them. But type 1 is the most common type. This is the most common. And basically what happens here is you contact some allergen, something in the air or something that causes some kind of allergic reaction in your body. When you first contact this allergen, nothing is going to happen. But the stem cell of antibodies accumulates mast cells and basophils. And when you meet this antigen the second time, you're going to get an antibody response, and that stem cell is going to release these basophils and mast cells, and they contain histamine.
Yep.
It's going to cause vasodilation, it's going to cause some localized inflammation, and this is what you get. There's going to be a lot of outpouring of fluid from capillaries, a lot of mucus production, and sometimes, if it's pulmonary, you can get an asthma-like effect from this too.
Why is it how is it you can develop an allergy to something later in life? All of a sudden I'm allergic to things that never bothered me my whole life. How is that? That just all of a sudden something that's never bothered you bothers you?
You know, I don't know, but it's happened to me too.
Okay.
For example, I'm allergic to formaldehyde.
Oh Lord, you're in the wrong line of work then.
Interesting.
I've been around it for years and years and years. And one summer I was teaching a human anatomy course at Rebecca Nazarene University in Nashville.
Mm-hmm.
So we had cadavers, human cadavers.
Mm-hmm.
I had my own cadaver, and I would prosect this cadaver to show the students what they should be finding on their cadavers.
Right.
And I noticed my hands broke out. Bad.
Big
I
case
thought
of
I had
hives.
latex allergy.
Uh-huh.
So I treated with steroids. I got some white gloves.
Yeah.
I put those on first, then I put on my other gloves so it wouldn't be touching my skin. And eventually it got better. But see, I only taught it for the summer.
Okay.
So I got away from the formaldehyde. So I come here
Mm-hmm.
and I'm dealing with this formaldehyde and my hands break out again. So I went to the dermatologist.
Mm-hmm.
And I wasn't in the room very long. He was very perceptive. He asked me a couple of questions. He looked at my hands and said, "You're allergic to formaldehyde." I said, "I thought it was a latex allergy." He said, "No, latex allergy is on the dorsum of the hand, typically right here."
Okay.
But my hands broke out terrible. But I was never I never had this problem before. So I don't know why. I may just
Just happens. Just old age, we're all
I don't
getting
have an
ruined. Okay.
Yeah.
Okay.
All right, so most of the time in type 1, it's a localized reaction. And depends on, if it's respiratory, you get runny nose, itching, watery eyes, and so forth. If it's GI, you get nausea, vomiting, diarrhea. All right? And if it's respiratory, you might get also asthma symptoms. All right? Treated with antihistamines, that's common treatment. Sometimes it's possible not to have a localized reaction, but to have a systemic reaction. So you get a massive outpouring of histamine. And what does histamine do? It's a vasodilator. It produces massive peripheral vasodilation. What's that going to do to your blood pressure? Drop it, all right? And it can also cause bronchiolar constriction, called respiratory distress. You can die from this. This is an anaphylactic reaction.
Okay.
So, for example, people who are allergic to bee stings sometimes carry a syringe of epinephrine with them. What is epinephrine? It's an adrenergic drug, isn't it? It causes peripheral vasoconstriction, stimulates the heart, and it dilates bronchioles.
Just to go back to the anaphylactic, if you don't avoid, let's say a bee sting is, here or there, but let's say you have an allergen, and for people who don't stay away from the allergen, can that systemic reaction become worse and worse and worse after they keep, no, okay.
I thought that leads to asthma. Like kids who, or people who are, allergic to certain things and they continue to be around them, and eventually leads to asthma. That's why.
Not sure about that. All right, this is another type of hypersensitivity. It's called subacute. I won't go into all the details, but some examples of where you would see this are in such things as systemic lupus and rheumatoid arthritis. Now, we're talking about antibodies in the class 1 that I just showed you. We're talking about antibodies here, so those would be B cells, right? This one here is T cells. It's cell-mediated. It involves the helper T cells and sometimes the cytotoxics. What happens here, we're dealing with haptens. Remember what we said haptens were? Haptens were incomplete antigens. In other words, it's something that by itself would not be a problem to you, but if it combines with one of your body's proteins, it becomes, it's perceived by your immune system as foreign, so you get a reaction but not immunity. So that's what this is. And so here are some examples. So you get various types of allergic contact dermatitis. So, for example, formaldehyde, sensitivity, poison ivy. Anybody ever had poison ivy? Oh, it's miserable. I had it when I was a kid. It is miserable. All right, certain cosmetics can do this. Tuberculosis skin tests are this type of sensitivity right here. All right. Well, I kind of rushed through that, but you got detailed notes. That's why they're so detailed, because it's sometimes hard to get a handle on this. You've got review questions, and you got